Hello everyone, it's very glad to have this opportunity to give a presentation here. The presentation topic is the tomography-based DEM simulations and machine learning-based constitutive modeling of sets. My name is XIONG, and my supervisor is Professor Jianfeng Wang. I come from the Department of Architecture and Civil Engineering, City University of Hong Kong. This presentation is divided into the following parts. In the first part, I will briefly introduce the background of the research. In the second part, I will show you the experimental devices X-ray microcity. And in the third part, I will show you how to reconstruct sand particles and to conduct morphological gene mutation. Then, based on all these techniques, we will generate a large DEM database. Based on this database, we will complete the data-driven constitutive modeling of granular soils. Lastly, is the conclusion remark. Sands with different geological origins exhibit highly different particle morphologies and microstructure. The different particle morphology and microstructure may have great effects on the micro-macro mechanical behaviors of granular soils, and that's what we concerned and want to investigate. The first part, X-ray microcity investigations. First, let me introduce the apparatus adopted in this research. You can see that the sample utilized is a miniature one with 16 mm in height and 8 mm in diameter. This is a picture of the experiment platform in the Shanghai cyclotron radiation facility. It's composed of X-ray beams, rotation, stage, and a detector. Our device for triaxial test is placed on the rotation stage. Note that the energy utilized is 25 keV and the detector has a spatial resolution of 6.5 micrometer. Here is a video to show the macro scale behaviors like stress-strain responses, the shear band evolution as well as the grain scale behaviors such as the translation, rotation, and the branch vector of both gained and lost contexts. As can be seen from the picture, each particle in the 1-to-1 mapping sample is numerical twin of the physical particle from the X-ray microcity experiment, and the softball membrane technique is adopted to simulate the latex membrane utilized in the experiment. And this technique is mainly used to calibrate the model parameters. The following figures shows the stress strain and volumetric strain curves after calibration, and we can see a good agreement of both stress strain curves and volumetric response reflects the feasibility and capability of this 1-to-1 mapping technique, and further validate the model parameters utilized in this study. And here are the comparison of the fabric evolution also good result. Then comes the tomography-based additive reconstruction of sand particles and morphological gene mutation. First, we need to introduce the morphological gene for granular particles. Through spherical harmonic analysis, each micro-CT reconstructed particle can be represented by a series of SH coefficients. These SH coefficients contain all the morphological information at different length scales, and hence can be treated as the morphological gene. Any particle within a given sample can be seen as a variance from the average particle of the whole sample. So each particle in the given sample can be expressed by the following equation 5. Well, this one, dUB and the sigma b square reflect the average particle surface and the overall variance from the average one for a given sample. Then, the spherical harmonic-based pre-symbol component analysis performed on the variance matrix. First, we set all the covariances of SH coefficients at different frequencies to 0. Then, the obtained matrix is symmetrized by its transposed matrix. After that, the pre-simple component analysis is performed, and any particle within the given sample can be reconstructed by the last equation. We then adopt the traditional morphology descriptor, targeting at different length scales to visualize this reconstruction technique. For instance, aspect ratio is for general form, roundness for local roundness, and the last 3 are the overall shape parameters that can be independently influenced by different length scales. We can see that the additive feature of the reconstruction technique and the 1 to 20 pieces are made for general form, 31 to 73 pieces for local roundness, and 74 to 91 pieces are for surface texture. The morphological gene mutation can be successfully implemented by varying particle morphology at specific length scales by the scaling factors. The following figures visualize the morphological gene mutation here. We can see we can successfully implement morphological gene mutation at the general form as well as the local roundness. Then comes to the generation of a large DEM database based on the micro-CT-based DEM. So first, we extract individual particles from the CT slices and then incorporated those particles into the spherical harmonic-based pre-symbol component analysis to conduct the morphological gene mutation at different length scales. After that, we incorporate all these particles into the DEM simulations to get the result. So totally, we create a large database that contains 10 groups of DEM simulations. Note that the first 1-to-1 mapping sample is only utilized to calibrate the model parameters and will not be used in the model training and testing. Therefore, a large DEM database comprising 200 and 17 DEM simulations of morphologically gene-mutated and gene-decayed samples was generated by the microcity-based DEM technique. Then consider the data-driven constitutive modeling of granular soils. We use 3 algorithms. The first one is a background neural network. The second one is a long short-term memory neural network. The third one is the gate recurrent neural network. We can see all these 3 models show almost equally good performance in capturing the main characteristics of granular soils subjected to monotonic triaxial loading, such as the effects of particle morphology at different length scales, sample initial packing state, and confining stress conditions. This result implies that the structure of micro-macro datasets of the granular samples and the monotonic triaxial sharing is relatively simple, so the advantage of LSTM and GIU is handling long sequence data is not well reflected. And then we compared our prediction performance with the microcity experiment. All the 3 model predictions agree well with the microcity experimental results, reflecting the feasibility and capability of the adopting this algorithm to model granular soils. And the last one is the conclusions. This study adopts a microcity-based DEM technique to generate a database for machine learning-based soil prediction models. This technique includes particle morphology extraction by X-ray microcity, morphologic reconstruction, and gene mutation by spherical harmonic-based pre-simple component analysis, and DEM simulations of triangle tests. A large DEM database comprising 217 DEM simulations of morphologically gene-mutated and gene-decayed samples were generated. Based on this database, 3 widely utilized neural networks with the capability of multiple outputs, namely BPNN, LSTM, and GIU, were adopted to data-driven constitutive modeling of granular soils. In these models, the deviatoric stress and volumetric strain were the output, while the other parameters, elongation, aspect ratio, roundness, sphericity, ratio of volume to the surface area, convexity, initial weight ratio, mean particle volume, uniformity coefficients, confining stress, and axial strain were the input parameters. All 3 trained models can reasonably predict the deviatoric stress and volumetric strain and the whole range of axial strain, regardless of the particle morphology, initial packing state, and stress condition. The excellent training and testing results reflect the feasibility and generalization of these algorithms to investigate the data-driven constitutive modeling of granular soils. Among all these models, LSTM and GIU outperform BPNN in predicting the deviatoric stress volumetric strain axial strain relationship of granular soils. With more local information, with fewer weights and biases, more efficient computation, and more stable and even error distribution for different stages of axial strains, GIU show a better predictive performance than LSTM. That's all. Thank you for your attention.